This is a very important discussion. Thank you very much. So this is a very important discussion, and I want to make sure that we go through this. I have been getting these questions for some time and hope that is exactly correct. What would happen? So this morning when I was responding to Bernie, I was saying, what happens if the SARS-CoV stays for longer and infects a larger set of population? Will we find people who may not be infected? It may be that eventually all of us are infected, or a majority of us are infected. And it may be that you can't find youngsters who do not have SARS-CoV-2. And youngsters may not even know, or young individuals may not even know that they were infected and have recovered, and they may have antibodies. So I think there may be some adjustment to this decision that Army has made. So back here, second topic for today. Are there multiple strains of the SARS-CoV-2? And more importantly, are there two separate strains, one strain which is more lethal than other strains? So there is a study I'm going to show you which has claimed that there are two main type of strains for SARS-CoV-2, and one is more lethal than other. And I'll also show you the study that says, no, this is not correct. And then I'm going to start from here. Look, there is a site. We have looked at that site in the past as well. That site is called Next Strain. So if I can have this refresh. So this is the nextstrain.org. Over here, they follow the strains of a virus. So what happens is, and look at this whole thing is SARS-CoV-2 virus strains or mutations in it since December, or maybe a little bit before as well. And what they have been doing is they have been tracking, they've been receiving data from various countries, scientists and researchers who have been sending them data, and they have only presented 3,000 strains here. They say that we have thousands of more strains that we cannot publish here just because our tool does not have the capability of handling with good performance more than 3,000 strains. So here we have looked at this in the past as well. This is all mutations of this SARS-CoV-2. So if I play this, pay attention that you would see that in here in China, there will be the outbreak, and then the outbreak would start spreading, and every color is a different strain, or there is a mutation. So check this out. If I play it, so here in China it starts, and it is very slowly moving. I hope I can accelerate it. Anyways, I think it's going to cause too much time. But generally what you would see is that as it starts migrating from China to other countries, even within China, the virus is going to start mutating. So question is, why is it mutating? So viruses tend to mutate, number one. Number two, keep in mind that viruses mutate to adapt to their host in which they are now living. So this virus is new for us, but it is trying to be our friend. It is trying to adapt to us. So I'm going to stop this playback, but just see that here we have some more strains that are evolving here in the US, then in the Europe, and then there is cross contamination where people are traveling from one place to another and they are bringing one kind of a strain to the other and so on. Apologies for my cough. I have allergies, and nowadays there are too many pollen out there. As you can see that the strains are they are being brought in from various people in one region to other. And if you go over any one of the strains, they would tell you, for example, this one, USA, New Jersey, nucleotide mutation. Nucleotide is a single nucleotide, right? Base. We'll talk about it in a second. Amino acid mutation as well. So amino acid mutation will mean 3 nucleotides are mutated together, correct? Because an amino acid is marked by 3 nucleotides. Divergence, that how much is the change, and then the date of the change report and the country and the author who found it. So you can actually just hover your mouse over this and see that there are changes. And these are 3,000 changes here. And they say they have a lot more changes. Now the question is, are these changes dangerous? The most common question that I've received from the students or from our viewers, so again, in our viewers, there are medical students and nursing students as well, so I call them students. But of course, there are other viewers as well. So the most common question that I've received is that when we make a vaccine and the virus is changing, will the vaccine become useless? So that is what we are going to have a look at today. So let's see it. And tomorrow we will talk about the monoclonal antibodies. And if we got time, we'll talk about cancers and SARS-CoV as well. So let's look at this discussion for a second. Look, are there multiple strains, or I would say mutations? Answer is yes. Most of these mutations in this study here, so they are the ones that I'm using here. They have pointed out about 198 important mutations. They are saying that the most of the mutations that are occurring are mostly for this virus neutral, or maybe deleterious to the virus itself, that the virus makes a change and then that change is just useless for it and it becomes even less useful or viable. And it is trying to live with us. So it is trying to change some of its proteins to adapt to host environment or human environment. Because just like it is new for us, we are new for it as well. And just like we are trying to adapt to it and figure out how to work with it, it is trying to work with us as well. So let's look at how that is happening. So Riyadh says, we don't want to be a friend with this virus. Just tell him, please, doctor. I would tell him, but he wants to be our friend at this time. So Cat Lady says that was the best virus drawing ever. Which one? The drawing which is right here? Okay. If that's true, then he would have been infected between December 14 and December 23. So we'll talk about that as well. That when was this? UK patient had been quite ill December 23. They didn't know. So let's talk about that as well. We'll cover that in the same study. So actually, let's just answer that question first of all. So there have been many, many messages that it is possible that SARS-CoV-2 was out there even before December. And this study, what they did was, of course, they are pulling the data from various countries and getting the virus genome and then comparing those genomes. And every genome that is sent to them has a date on it. So what they have done is they've said that in humans, this genome is observed between 6 October and 11 December, the very first strains of it. So they say that it cannot be before that time. So if you have any thought that it is between October, November, December timeframe, then that is correct. But if you feel that it is September or August or before, then that is not correct. So that means that it is possible that some people were sick before December. What I cannot say is that, is it possible that the virus was outside of China before December? I can't say that, but maybe it was possible. I don't know. So back here, number one, are mutations possible? Yes. Are mutations happening right now? Yes. The mutations are, what are those mutation viruses trying to live with us? Now let's see where are the main mutations. So this is the viral genome here. This is the genome or nuclear material of the virus. And if you see here, this is open reading frame 1A. Then I'm going to make it a little big here. Open reading frame 1B. Then S, which is the S protein making gene. This gene, when it is translated, it makes this S protein of the virus. Remember the spike protein which connects with the ACE2? Then there is open reading frame 3A and 3B. Then there is E protein creating gene, that is this one here. Then M protein creating gene. So these proteins are in the viral membrane. So here is the, let's say E, and this is M. This is S. Then there is open reading frame 6, then 7A and 7B, then 8A and 8B, and then there is N protein, this guy, the one in green, N protein is a protein that makes that makes a capsid around the RNA, and RNA and the protein together are called nucleocapsid. So N protein making gene, and then there is NB, and there are many other genes as well. This virus has a very large RNA. In this, I have highlighted that according to the study, the majority of the mutations are occurring here in the open reading frame 1A in a part of it, and they have actually given the number of that part. And then in the S protein, which could be unfortunate, because if the virus started making changes to its S protein, which binds to our receptor, it could be unfortunate for the virus or for us. If the virus ended up with the protein, its own spike, becoming D-shaped, and it could not bind with our ACE2, then the virus will become harmless to us. And maybe there are such mutations, and maybe those viruses could not harm us anymore, and they just died. But there is a change going on, mutation going on in the S protein as well. What they're saying is, fortunate for the virus, those changes are mostly outside of the S protein itself gene and just surroundings of that gene, or part of before the end terminus, in more technical terms, before the end terminus and before the end of the protein. Then there are changes in the open reading frame 3A as well here. So these are the areas of greater changes. What is the significance of that? I just discussed about the S protein. Luckily for the virus, those changes in the S protein are not significant enough or in the right positions to change the S protein shape and to make the virus not able to connect with us. So virus is still connecting with us. So these mutations are still not damaging to the virus, and they are not helping us by changing the shape of S protein. So that's S protein. In the open reading frame, this side, 1A, they are saying that the gene that is mutating, that gene is antigenic. Antigenic means that there is some protein formed by that gene, which our cells detect and say, I don't like this. This seems like something from outside, and I'm going to react to it. So it is antigenic. And what they're seeing is that this protein that is antigenic and is mutating is causing CD4 and CD8 cells in our body to react against this virus. So maybe the virus is trying to adapt to our body to see if it doesn't want to cause our immune system cells to react to it. So it is trying to change there. So what is happening is, I think we all know, so let's do this. Let's say here is the coronavirus. We do this immunology discussion every day almost. So now you must be very expert in this one. So here is a coronavirus. Usually macrophage or dendritic cell or some antigen presenting cell eats it up. Then they present it on their surface. A part of this. So today this is very, very important. Hey, Alexandros, Vasaras, doctor, welcome. So this is very important. So look, and this would be important tomorrow as well for monoclonal antibodies that we would discuss. Let's say this is a virus. This is the virus. And I'm going to intentionally make it in multiple colors. So why I did that was, the virus has various patterns on it. These are various patterns. S protein has a pattern. Let's say E protein has a pattern. M protein has a pattern. The cell membrane has patterns. Inside, the antigens have patterns. Our body does not recognize a complete virus. What it does is it chops up this virus. So if I take a, what is this thing called? An axe-like thing. My wife must know what it is called in the kitchen. And if we chop up this virus into smaller pieces and we lay them straight, then we can have our cells that may bind to parts of this material. So we may have a macrophage that might recognize just this part or just this part or just this part. So this is what we say. Yeah, cleaver. Thank you, Gazar. This is a cleaver. Thank you, Evelyn. And thank you, Kenneth. So many helpful folks out there. So yeah, so if we take a cleaver, in our case, that cleaver is a macrophage, butcher knife as well. Yes. So in our body's case, that cleaver is going to be this macrophage or neutrophil or dendritic cell. So these are the ones that take up the virus and break it down into smaller pieces. Then those smaller pieces are shown on their surface. And then what happens is, so let's take one piece here. Let's say we take this one piece and we say that the shape of this piece is like this. This is the shape of this piece. This is a protein, part of the protein, very small part, 7-8 bases, 7-8 nucleotides. So not nucleotides, 7-8 amino acids. It's a small part of the protein. And this is the pattern on it. Now let's say that the macrophage is displaying that part of the virus on its MHC2 receptor. Oh, sorry, not receptor, the MHC2 protein. Then comes a T cell, for example, and the T cell has to have a receptor that can bind with this shape. And now if you ask that, hey, how come we have a cell that may have a shape that can bind with it? Our immune system is made in a way that when those cells are produced, every cell in a random way gets a new shape with which it can bind. So B cells and T cells have those shapes, receptors on them. So there are tons of T cells sitting. Some T cell have a shape like this, and they can have a box attached to them. Another T cell may have a receptor with a shape like this. Another T cell may have a receptor with a shape like this. So only the correct shape can bind with them or attach to them. And they're all sitting around. This is the same case for the B cells as well. They're just sitting around. And here is a question for you. What is a receptor, antigen receptor for a B cell? We know that for a T cell, these are T cell receptors. What is an antigen receptor on a B cell? Cat lady says she just watched for the drawings, but even in medical school or medical industry. You're very welcome. More importantly, we would share this information together and become more knowledgeable. I think that when we discuss these things together in depth, we become protected from the rumor mill and incorrect messaging from others. Okay, back here. So the question that I had said was, what is the receptor on a B cell? Remember this, that B cell receptor is usually immunoglobulin D. Immunoglobulin D is the receptor. It acts as a receptor on a B cell. So B cell, you're most welcome, cat lady. So B cell makes the immunoglobulin D, and that is what gets on its surface. And that immunoglobulin D, if I go to the binding site of this immunoglobulin, this would also have similar binding shapes. And only antigens that can bind with that shape or that can fit into that shape will connect with this B cell. And every B cell or a T cell can only bind with one kind of a shape. So that means if we say in nature, there may be antigens which are one, I'm just making up, maybe there are a billion, then we need at least a billion B cells or T cells with those shapes. This is why, so I don't want to cover the whole immunology here, but this is why we try to bind with a smaller size of the protein so that the shapes are lesser. If we try to connect with the larger proteins, and larger proteins are in billions and billions of shapes, and we cannot do much about it. So back here, now we know that the B cells also have receptors on them. T cell also have receptors on them. So once the virus comes in, let's say coronavirus, our macrophage has eaten it up. Then macrophage is now, it has chopped it up into smaller pieces. One of those pieces sitting out here on the T cell now, and it kind of binds with the T cell. Then there is co-stimulation, both chemical and receptors, or sorry, binding molecules. When the co-stimulation occurs, then the T cell is going to release, let's say, interleukin 4, which causes the B cell to become active. Now, which B cell would become active is a B cell. So there are going to be thousands and thousands of B cells sitting here as well. They would all not become active. Only that B cell that has its receptor, which is primed with a virus. So let's say this was coronavirus, and that B cell was binding to that shape as well. Only those B cells that were now attached to some antigen and they got interleukin 4, these B cells will become active. And what will they do? They will start making immunoglobulins. What will be the shape of the immunoglobulin? The same shape as the IgD here. So when the immunoglobulin is released from these B cells, I'm going to make the shape here, and that shape is going to be similar shape. So this is very important for tomorrow's discussion as well. This B cell antibody, when it is released into the plasma, it is going to go and find viruses wherever they are sitting. And as long as they have this pattern on them, or pattern, pattern on them, they are going to bind with that virus and kind of make it really, really sad, because they are going to neutralize it. So this is the way our immune system works. Today we went a little bit more in depth about the shape of the connection, or we call it epitope. Epitope. It is the part of an antigen that is recognized by our T cell or B cells receptors, or even presenting cells receptors. Okay, back here. This open reading frame 1A, the changes that are happening here. The antigen that is made, the protein that is made by this gene, has a pattern that allows CD4 and CD8 both cells to become active. What does that mean? That means not only CD4, so this is the CD4 cell here, and then CD4 cell is going to make sure that the B cells are activated, but then CD4 can also cause, it can become T helper 1, and that would cause the cytotoxic T cell, T cytotoxic, to become active as well. So this is CD8 plus, this is CD4 plus. So the antigen in the virus is trying to change itself. Maybe it is trying to evade this system so that the shape of the pattern of the antigen is changed from here. So instead of this shape on this place, maybe the shape becomes like this of the antigen, this area. And when the shape is like this, then our immune system is not going to react anymore. So that is how the virus is trying to adapt. So back here, what they have seen is they have seen 198, if I can get your attention on this side, 198 areas of change in the virus within this time frame. This is October till now, so not very long duration. And they've seen a lot of changes. Those changes are the open reading 3A frame also is making a change to the CD4 epitope, which means that the pattern that can activate CD4 cells, it is trying to change that as well, the virus. There are changes to the S protein area as well, and that is what's happening. So the outcome of that, so from this study, I would recommend that you go to the bottom of the study, and they have a few discussion points that are interesting. I have highlighted them here for myself, but so last 3-4 paragraphs for discussion, I'm going to just present them to you directly, but you can read up. What they're saying is this: from looking at these patterns of the virus, or changing virus, number one, they feel that it started between October 6th to December 11, and not before that. So that is one. This virus was born in this time. Not born, it jumped into humans at this time. Secondly, they feel that this virus is trying to, and every virus does that, it is trying to adapt to our body in a positive way, trying to become our friend and stay with us. So the most of the changes are in that direction. Then here is the basic problem. Let's say you are a vaccine manufacturer, or you are going to make a drug, let's say monoclonal antibody that we'll talk about tomorrow. Do you think that if you made a drug or vaccine that targets the proteins that are generated by the areas that are changing, would that be a good idea? So if I made a vaccine, or I made a monoclonal antibody that reacts against the epitope produced by this area of hypermutation or mutation, would that be a good idea? No, because the virus is changing it. So vaccine may work today and may not work tomorrow because that area has become changed. So the reason for studying the changes in the virus are not only to say that is the virus becoming more lethal or less lethal. Normally viruses become less lethal as they mutate and they adapt to us. But this is also important to make sure that the scientists make vaccines and monoclonal antibodies against stable parts of the virus. Those parts that are not changing very frequently and not changing very much. This is why this data is very important. So that means if you and us, we are together researchers, we will say, hey guys, do not target open reading frame 1A area 3600 something, or do not target the S proteins area around it, or do not target 3A and so on. And by the way, S protein itself is a very big target right now because it is itself not mutating. The area around that in this gene is mutating, but the S protein is not mutating. But we'll say, hey, don't do anything with the 3A, don't target that. So don't make a vaccine that goes after 3A or so that is the point. So back here, vaccine evasion. Vaccine evasion means that the virus may be able to evade a vaccine if it changes. So we have to make a vaccine that targets its stable area. Similarly, drug resistance. If a drug starts targeting an area that is mutating faster than other areas, then the drug will become useless after some time. So because of that, it is important to learn this. Now, the last point for this virus is this one. So there is another study. Let me show you that study. So here, this is a study done by Tang et al. And I also want to show you that there is a counter study or an analysis of their study by McLean et al. Tang et al. Said that they have looked at 103, I believe, 70, 30, 101. So they took a number of cases and took the virus from them, the genome of the virus. And Riyadh, I'll answer you in a second. So they took the virus genome and they looked at those genome patterns. And what they found was that there were two parts of the virus that were different from each other. So let's say part A and the part B. So they found that there is a cluster of viruses that has part A, which is more prevalent in them, or genes or proteins which are more prevalent. And they saw another strain or species of virus. I shouldn't call it a species, another strain of this virus, which has a cluster of change in this area. And what they said was that as they did the analysis, they found out that the strain that they called S strain, that strain had come in first. There were 29 viruses of that genomic pattern of SARS-CoV-2. And then they said there were 72 viruses of a slightly different genomic pattern. And from this, There are many other smaller changes as well. As you just saw in this study that I just showed you, they have found 198 changes. So if you see here somewhere at the top, let's see, 198 sites that are being changing. So it's not that there is just two sites. What they're saying is, Tang et al., they said that there are two kind of categories of sites that were more important in when we compare the strains to each other. And based on that, they said that we have one strain of virus that can be called an S virus. That was the ancestral. So because of that, S for ancestor, and I made this virus have a little beard as well. So this was the original virus that started in Wuhan. And then as the virus started spreading, the mutation occurred and another predominant category came in, which they called L virus. So L for child, for example. So the L is child. So you can remember one of these, L for child, or you can remember S for ancestor, and then the other one is the other one. And they said that the child one, or the L category, had 72 counts. From this, they concluded that because the prevalence or the number of L type was more, that must mean that it was more lethal. That must mean that it was spreading more, surviving more, and infecting more, and was more contagious, and so on, compared to S, which could not spread with that as much frequency, with that as much strength, and it probably was going into people and dying there. So the survival of the fittest, they saw that the L was spreading more. Based on that, they said, you know what? Yeah, so the S was the senior. S for senior, yes, that's a good one. And the L for child. So based on this, they said that the L type is more lethal than the S type. Now, I also want to show you a counter study here. This was done by McLean et al. And they said that we looked at the same data that Tang et al. Looked at, and we feel that they are incorrect in their analysis. And it is possible, guys, this is a rapidly changing situation. We should be grateful to Tang et al. For what they did and try to say that, hey, there may be a more lethal strain coming up. And we should be thankful to McLean et al. As well for going in again and looking at that data and saying, hey, here are some adjustments to your stance. So the adjustments that are important, they have looked at claims, the first claim and then the second claim. And I would give you these as usual. I have these links. I'll put them in the description of this after this talk is over. So generally what they've said is, they've said that, look, just because in 101 people sample, we saw that 72 were of type 1 aggregated change type and 29 were another, means nothing. Maybe you would go to another hundreds cluster and you might find S to be more than L. So they have alpha child. Yes. So based on that, they feel that it is not necessarily accurate to say that in our 100 samples, whichever is more should mean that it is more lethal as well. Maybe we just by chance happen to get that cluster. Maybe you go to a different place and you would have a different type. So that is one. Secondly, they have done a more rigorous analysis of the data as well, which I actually did not have a chance to go and look at. But their second claim was, compare the frequencies of non-synonymous and synonymous substitutions in the data, claiming that there is significant evidence of selection suppression of the frequency. So what they're saying is that in the data, Tang et al. Are saying that there are certain parts of the genes that are being suppressed or not used. And that may be because virus is trying to select us by changing its genome in a way that it can adapt to us. And they are saying that they looked at that data and that also does not hold up correctly. So in my opinion, after looking at both the studies, the whole conclusion of the discussion will then become, yes, the virus is changing. Yes, the people who are making vaccines and the making antibodies, monoclonal antibodies, they will have to make sure that they target them to the area which is not changing very fast. Yes, the virus is trying to adapt to us. If it did adapt, it will become harmless instead of harmful. Viruses want us not to die so that they can live in us and enjoy their life. The idea of two strains of S and L are actually, I think, not correct. So I tend to side with McLean's et al. Study to say this data and this inference of the data on Tang et al. Does not look right. Because you just take 100 people and whichever, some virus is going to be more in them, you cannot all of a sudden say that this is the more lethal one. So that is the discussion for today. I wanted to go over the monoclonal antibodies as well, but it looks like we don't have sufficient time today. We'll have that discussion tomorrow. I'm going to try to do the monoclonal and the vaccines. Plus, we'll see if it's as well, SARS-CoV. And as you saw, I very smartly did not name this video with SARS-CoV at all. But as I post the links, maybe the AI is going to pick up that as well. We'll see what happens. But thank you very much for all your support. And please put your comments in the YouTube or Facebook. I usually go and answer all comments. I'll spend next 2-3 hours answering the comments. So please have your comments there. I'll answer them. And then we'll go from there. Tomorrow, monoclonal antibodies, which is a very important topic. Have a good day. And thank you very much. I've gotten so much support that you cannot imagine. I've gotten so many comments, so many heartwarming comments to ask us to keep going. And guys, I'll tell you this, I have never here said that I have a website as well, which is our business, drbeen.com. And I wanted that this work stays free for all of you because we need it all. So there is some issue now, but I'm going to continue to try to put that work out here. There has been suggestion for putting it on some other areas as well where it can survive better. So I'll do that as well. The reason for me to put that on YouTube or Facebook is to keep it free. Our site has a fee structure with it, and I don't know how to make it free there. So I was putting those in here so that it stays free. But as they're taking it down, I'm going to figure out what to do. So once again, thank you very much. Let's keep becoming smart about SARS-CoV-2 and the COVID-19. I think it helps all of us stay protected from rumors and be able to think correctly. So talk to you tomorrow.